Hello, and welcome to another video in our series about the four-process model where we explain the basic biology of aging. This one is about age-related diseases. I am Dr. James Wordsworth, shown here on an ice rink, gracefully coming to a stop using my face and body on an unsuspecting skater. So let's talk about how aging causes disease. First of all, it's important to remind ourselves that aging is not simply the passage of time. It is a specific process, and it is not increasing robustness, robustness, and it is not increasing robustness, but reducing robustness, leading to age-related diseases. So to put this another way, aging and diseases are one. They are the same. It's not that aging increases the probability that other things will cause diseases. Age-related diseases are a direct result of aging. They are part of the same process. So in the old model, we had DNA damage leading to things like inflammation and cell senescence, and we had a bunch of positive feedbacks leading to more damage, more senescence, more inflammation, and this led to cell death, cell loss, damage accumulation, inflammaging, and that was supposed to cause age-related disease. I'm going to explain this in more detail in more videos to come, but essentially these models didn't work. They are absolutely terrible at predicting how and why we get age-related diseases, which is why there are almost no models for how we get age-related diseases as a direct result of aging. And that is because when we actually test these things experimentally, often as not, we find that reducing DNA damage accumulation decreases lifespan, or increasing damage accumulation increases lifespan, and there's no real relation between how much damage a person or an animal accumulates and how long they live in almost every experiment you can name. Mitotic organs generally don't show any cell loss, and anti-inflammatories often as not exacerbate disease as they do improve it. Generally, there is no relation between this old model and what we'd expect to find when we test it experimentally. But all of this changes when we start to look at the four-process model. Now, we talked about what the four-process model is in our introductory video, What is Aging, according to the four-process model. And those of you that have watched that may recognize this picture. If you haven't watched that, I would honestly recommend watching What is Aging before you watch this video. However, we are going to cover some of it in basic detail, but the idea is that these three processes of the four-process model, intrinsic aging, sclerosis, and extrinsic aging, will result in these different age-related diseases. And that's why we've created this Venn diagram to show which processes contribute to which diseases. So let's start with sclerosis. This is the idea that in organs where things have gone wrong, we've got lots of damage and cell death, we have this process of compensatory proliferation, which leads to diseases like fibrosis and cancer. And we covered this very basically in What is Aging, but we didn't really cover what these diseases were. So let's start with fibrosis. Now, fibroblasts are our cells which produce collagen and elastin and all the proteins that we have outside of our cells, called the extracellular matrix. And this extracellular matrix, or ECM, is highly important for allowing cells to sort of communicate with each other, to understand where they are. It's basically like a scaffold which allows a cell to move and survive. Without the ECM, our tissues are considerably less healthy, and the fibroblasts are the cells responsible for producing this ECM. And this is most important when we have a wound. When we have a wound, our fibroblasts enter an activated state, they proliferate, and then they produce a new collagen matrix, which allows the wound to close, and then these cells die off when the wound has closed. So how is this process regulated? So when we have a wound, we get an influx of inflammatory cells called macrophages, and the macrophages will activate the fibroblasts, and the fibroblasts will activate the macrophages. And the fibroblasts, crucially, also have a little weaker signal, which is self-activatory. So this whole process is a system of positive feedback, which due to the presence of the macrophages means we get fibroblast activation and proliferation. And then when the wound closes, the inflammatory signal dies, the macrophages leave, and they stop activating the fibroblasts. The fibroblasts obviously can no longer activate the macrophages. And the weaker self-activatory signal is not sufficient to maintain the fibroblasts, so we go back, once the wound is closed, to having a normal inactive fibroblasts or less active fibroblasts in our repaired ECM. However, the problem is this self-activatory signal, this weak self-activatory signal. Because if at the point that the wound closes and we lose the macrophages, there are already enough fibroblasts that they can maintain themselves by this weak signal, then instead of dying off when the macrophages leave, they will just continue to activate themselves and the signal will get stronger and stronger and stronger, and this causes fibrosis. And crucially for the four-process model, when we have mutations making fibroblasts more responsive to growth signals and sclerosis selecting for these faster cells, then what we get are cells that not only respond better to their self-activatory signal, but they submit more signal to activate macrophages, and they respond better or more sensitively to the macrophage signal activating them, which means you will require fewer cells and a shorter time period where the wound remains open in order to produce this point where we get sufficient self-activatory signal that the fibroblasts don't die off when the wound is closed, which means we get an increased likelihood of fibrosis. And just to note, at the same time, when we have intrinsic aging, which is reducing their metabolic rate, reducing their response to these signals, we get the opposite. We get fibroblasts much less likely to respond excessively to these growth signals, which can lead to the opposite occurring with aging. We get these chronic wounds which just don't shut because the fibroblasts never produce enough collagen to close the wound. So, in our new model, we are suggesting that fibrosis, here shown as idiopathic pulmonary fibrosis, which is fibrosis in the young, are a result of sclerosis. And now let's move on to cancer. So if we have our wild-type cells shown here in pink, and we have our faster cells in blue and our slower cells in red, our faster cells will obviously be associated with a higher mutation rate, which is a direct cause of cancer. However, the most crucial thing we are suggesting here is that when you get this transformed cell, this cancer cell, which no longer responds to controls, if you were to get that transformation in a tissue which is full of very fast metabolizing and proliferating cells, then those cells are much less likely to notice that cancer cell and kill it in time. Whereas if you have a slower tissue where all the cells around this cancer cell, which has suddenly shed all of its controls and is now proliferating like mad, then that cell looks like a bull in a china shop. Those cells can very easily kill that cell. Whereas in these faster tissues, the cell goes unnoticed and is allowed to proliferate and evolve until it becomes cancer and very difficult to kill. So that is why cancer is also a result of sclerosis. Now, our second process that we discussed in What is Aging was intrinsic aging, which is where our slower cells identify cells around them that are metabolizing and growing faster, and they induce these epigenetic marks which slow them down. And this was intrinsic aging, and it led to metabolic slowdown. And the result of that, we suggested, was that all of these processes are reduced protein translation, transcription, DNA replication, and cell proliferation, which means that over time our tissues are going to be less responsive to these growth signals. They will proliferate less and they are more likely to die off, which leads to processes like sarcopenia, which is muscle loss, and dynapenia, which is muscle weakening. And in bone, we have a very similar thing happening. So bone is regulated by two main cell types, osteoblasts, which produce bone, and osteoclasts, which degrade bone. Now, evolution has produced a very clever system here, because our osteoblasts respond to IGF-mTOR growth signaling, and that causes them to produce more bone, whereas osteoclasts are inhibited by these same signals. So when we have intrinsic aging and cells that are less responsive to these signals, we get less response to IGF-mTOR, which means that our osteoblasts do less and our osteoclasts do more, which leads to osteoblast activity reduction and osteoclast activity increase, leading to osteoporosis. So osteoporosis and sarcopenia, we suggest, are direct results of intrinsic aging. Now, the third process we discussed in What is Aging was extrinsic aging, where due to the decline of all of these processes, we have reduced use of ATP, and therefore we produce less ATP, which restores our ATP-ADP balance, and that causes a buildup of the fuels that we would otherwise burn to produce ATP, so lipids and sugars. And because these things are toxic to the cells in too high amounts, our cells inhibit the import of these fuels, which means they reduce the insulin receptor, which is responsible for fuel import. Which means while we restore the balance in our cells, this is associated with insulin resistance, which is one of the main problems we have with aging. It also means that because we don't want these fuels building up in our blood, we store them in our adipose, our fat cells. So we gain weight with age, and worse than that, because our adipose homeostasis is maintained by inflammation, which attracts macrophages to remove these hypertrophic fat cells that are full of fat, we get chronic inflammation with age. And all of these factors contribute to extrinsic aging. However, we go further than that. We say that because we have these two different fat stores, the subcutaneous adipose and the visceral adipose, we also get visceral weight gain, metabolic syndrome, and type 2 diabetes when we have far too much of these excess fuels in our systems. So, extrinsic aging leads to metabolic syndrome and type 2 diabetes. Let's move on to cardiovascular disease, probably the main cause of death outside of cancer, in some places including cancer, or at some points in time including cancer. It was or is the largest cause of death. So obviously cardiovascular disease sounds like a disease of the heart and blood vessels. This is not actually true. It is much more specifically an arterial disease, and it has two major forms. We have the stiffening of the arteries creating high blood pressure, and we have the formation of plaques, which we call atherosclerosis, the formation of atherosclerotic plaques in our arteries. And the reason it's so associated with our cardiovascular system is because the coronary arteries, which feed our heart, have such high blood pressure, and they are frequently the sites of these plaques. That's why we call it cardiovascular disease. So atherosclerosis then is the formation of these fatty plaques in our arteries. So atherosclerosis is the formation of these fatty plaques in our blood vessel, which increase blood pressure because obviously they restrict blood flow. However, the direct inducers of mortality occur when these large plaques break away in our large blood vessels and they travel through our bloodstream until they get wedged in a smaller blood vessel where they no longer fit. And what this causes is the organs that this blood vessel is feeding don't get their necessary supply of oxygen and nutrients, and then they die. And or we get the bursting of the blood vessel behind the plaque because of the buildup of pressure. And that causes heart attacks in the heart, strokes in the brain, and pulmonary emboli in the lungs, which are some of the main age-related causes of death. So what does this mean then according to the four-process model? So if we look here at this young artery where we have the lumen or the main or the actual blood vessel with the flow of fat going through it, we have this protective glycocalyx barrier which prevents things getting into the blood vessel wall, and that is maintained on top of our endothelial cells, which are the main cells which protect the blood vessel. And then beneath them we have what's called the intima and the smooth muscle. According to extrinsic aging then, what happens when we get the buildup of these fuels is a lot of them are converted to cholesterol, which is the main lipid molecule carried in our blood. And possibly people are familiar with these molecules, low-density lipoprotein, or LDL, and very low-density lipoprotein, VLDL, which carry most of the lipid in our blood. And these molecules can be highly atherosclerotic. However, not on their own. Our glycocalyx barrier is highly protective. Our endothelial cells prevent these cells from getting into our blood vessel wall, except when we have intrinsic aging, which when our endothelial cells become less responsive to these endothelial growth signals, we get endothelial cell death, glycocalyx destruction, and what that means is the LDL and other processes that the LDL and other lipid carriers that would normally remain in the blood then have access to the intima of our artery walls. They can flow in, and possibly this is a necessary process for the healing of our endothelium, forming the fatty streak. But when you combine intrinsic and extrinsic aging, what you get is far too much of this lipid forming in the wall, which creates an atherosclerotic plaque. You get the macrophages, which are there, and they should be removing all of this fat, but instead there's too much for them. They form foam cells, they get lodged in the plaque, and the plaque becomes bigger and bigger and bigger. So what we're suggesting then is that cardiovascular disease is a direct result of both intrinsic and extrinsic aging combined. Let's move on to Alzheimer's disease. So Alzheimer's disease is well described to be a result of amyloid beta, which when we get the amyloid precursor protein, we cleave it and we form this amyloid beta peptide, which then forms Alzheimer's plaques or protein plaques. And these build up, and that causes Alzheimer's disease. That is our current model of how Alzheimer's disease happens. We get amyloid beta and the passage of time forming more and more plaques. So the reason we think this is that when we have mutations in our amyloid precursor protein processing, or actually the molecule itself, we get more plaques and early onset Alzheimer's disease. So early onset Alzheimer's disease is generally caused by mutations in these very select few proteins, and it's pretty much the same disease as late onset, it just happens much earlier. So the problem is that now we have many excellent drugs which are very capable of removing these amyloid plaques. And does this cure Alzheimer's disease? Not a jot. It does almost nothing. There are people trying to say it does lots or does something. Maybe it does do something, but it certainly doesn't do a lot. It doesn't do what we'd expect according to this model, where we remove plaques, and because these plaques have taken so much time to form, we should have significant healing of the brain. And there were so many people trying to say, oh, we just didn't do it early enough. We didn't do it in people that hadn't had significant brain death, so we did more and more trials. And in all of these trials, it suggested that it either wasn't early enough still, or the drugs don't work. So what we're suggesting here then is that Alzheimer's disease, whilst it might be the product of plaques, is not the result of gradual plaque accumulation. Just the same way that we've suggested that aging is not the result of a gradual accumulation in DNA damage. It's specific processes that cause homeostatic or equilibrium shifts. And these shifts occur because our cells are changing. They're either becoming faster or they're becoming slower. And what these homeostatic shifts could mean is that there is a higher equilibrium level of plaque determined by that equilibrium. So why is this true? Because plaques are obviously governed by homeostasis in our bodies the same way as everything else. They are determined by the rate of formation, which is the rate of amyloid beta being converted into plaques, and they are determined by the rate of degradation through autophagy and inflammation. So when we have an increase in amyloid beta formation, we respond with an increase in amyloid beta plaque degradation. The only way that we might get an increase in amyloid plaque is if we have an increase in formation or a decrease in degradation without the compensatory shift in the other. And then that should cause a shift in equilibrium level of plaque. So how might this be happening then with aging? So if we have a young brain where brain wants to do something, it wants to send a signal, that signal is the result of a protein, and that protein is produced by a process of translation at the ribosome, shown here, and that's done at a specific rate where we produce a certain number of molecules over time, and these molecules build up until we have enough molecules and the function that these proteins were produced for is complete. At that point, these molecules produce a process of negative feedback where they inhibit each other, they inhibit translation, and they inhibit their own activity. And part of this process is the process of autophagy, where these proteins are degraded. So this process happens over a certain length of time. And what this means is when we have intrinsic aging and the rate of translation is reduced, the rate of protein production is reduced. And as a result, it takes longer for the proteins to carry out their function. which means before they induce the negative feedback and autophagy, which means if we just look here at this orange molecule, which was produced first, we now have an increase in average protein lifespan, and we have a reduction in autophagy, simply because we require less autophagy per unit time because it takes longer for our cells to do things. So how does this reflect an increase in plaques then? So if we have an increase in protein lifespan, then obviously this increases the chance that they might aggregate, and also because we have reduced autophagy, it reduces the rate of protein degradation, which means we have an equilibrium shift for more plaques. We also have extrinsic aging, which means we have more lipids and fuels around, which might also increase the rate of protein aggregation, increasing the number of plaques further. So Alzheimer's disease then is the same as cardiovascular disease. It's the result of a combination of extrinsic and intrinsic aging. Now, what you might notice from here is the mysterious absence of inflammation in what I'm talking about. And I discussed that in considerably more detail in the talk on how do we stop aging, where I look at anti-inflammatories. But that is not to say that inflammation is not important in processes or diseases such as rheumatoid arthritis and fibrosis, both of which are a result of increasing inflammation. Rheumatoid arthritis is essentially an autoimmune disease. So if you have increased inflammation, then of course at the same time you have an increased chance of your immune cells becoming activated and targeting your own tissue. With fibrosis, you might well remember that our fibroblasts were activated by macrophages. So obviously if we have more inflammation, we have more macrophages and we have more fibroblast activation signal, and we have an increased chance of fibrosis. So both of these processes are also a result of extrinsic aging through inflammation. So fibrosis is a result of both cellulite and extrinsic aging. What about cancer? So obviously when we have a process of chronic inflammation, that increases our chances of getting diseases like rheumatoid arthritis, where we have an overreaction of our immune system. In order to prevent this, we induce a system of immune tolerance. Our immune systems become less likely to be activated by signals because the signal is much higher. So what that means is we get less diseases like rheumatoid arthritis, but at the same time, when we get cancer cells forming in our tissue, where we would normally kill these quite easily, we now have a trade-off to be made. If we have a chronic inflamed tissue, we can either have the same degree of immune activation that we would have without the chronic inflammation, which means we get autoimmune disease, or instead we have chronic inflammation plus tolerance, where we have reduced likelihood to get autoimmune disease, but we have increased likelihood of getting diseases such as cancer, where we miss something. So cancer is also a result of inflammation and extrinsic aging. Now, we published all of this in our paper in bioRxives, Why We Age, the Four-Process Model. However, that is one of the longest written papers of all time, which is why I've also produced these videos. And for those of you who've noticed that I have mentioned only 3 processes of a 4-process model, I do have a video on the 4th process of the 4-process model. As I've described, we also have the introductory video, What is Aging, which describes the other 3 processes in more detail. And we have How Do We Stop Aging, which focuses on what the 4-process model means for anti-aging therapies. So we also have a podcast, Undoing Aging, where I interview leading scientists about the basic biology of aging. And all of this material can be found on agingrethink.com, or you could just subscribe to this YouTube channel to see what other content we have available. There will also be a book coming out at some point which will describe all of this in much more detail, but also intended to be more interesting and aimed at a wider audience without all the technical detail of the book-sized paper we published. So thank you very much for listening. I hope you learned something. And if you have any questions, you can email me at my Newcastle email address, james.wordsworth2@ncl.ac.uk. Thank you very much for listening. I hope you learned something.